Okay, thank you, Ben. Am I audible? First, can you hear me?
Yeah, you should be good.
Okay, yeah. Okay, thank you. First of all, I would like to thank Ben for giving me this wonderful opportunity to speak about my research in broader audience. My name is Abhishek Poddar, and I'm a fourth year PhD candidate in cell and molecular biology program at the University of Toledo. And today I'm going to speak about calcium spike regulates yeast cytokinesis. That's the title of my talk. So I'll start with how a cell divides. Cytokinesis is the final stage of cell division in which a mother cell physically separates into two daughter cells. So this crucial process is consists of at least three complex phases, which involves assembly of an actin and myosin based contractile ring. So it's assembly of a ring-like structure at the center of the cell. So as you can see, this center of the cell is called cleavage furrow, and this ring-like structure that is assembled at the center of the cell is called actomyosin ring because it is predominantly made up of two protein, actin and myosin. So hence it is called actomyosin ring. So this ring-like structure assembles at the center of the cell called cleavage furrow, and this ring-like structure undergoes constriction or its diameter starts reducing, and this ingresses the ring-like structure and pinching off the plasma membrane or the cell membrane with it. So this narrowing of diameter results in ingression of the or constriction of the actomyosin ring. And finally, the cell terminates by pinching off completely and the cell separates. It's a wonderful mechanism by most eukaryotic cells separate. This is how a normal human cell separate. But we do not work with humans in our lab. We work with yeast, which is a great model organism to study this similar process that is occurring in higher model organism like yeast, like humans. So why do we use yeast? So what is yeast first? So yeast, the specific species that we use in our lab is fission yeast, which is Schizosaccharomyces pombe, scientific name. It is a tiny single-celled rod-like microorganism which divides by medial fission, which means symmetrically from the center of the cells. So it is a great model organism to study cytokinesis, which is the final stage of cell division. But why it is a great model organism to study cytokinesis? Because its genome can be easily modified. That means the molecular cloning process is much easier in this model organism. So we can play with the genes that causes the mutation in whose causes diseases. And we can try to gain some insight of and the mechanism why it causes that diseases. And the mechanism of cytokinesis in this small model organism is very similar to yeast, very similar to humans. We can do quantitative microscopy in this model organism, and we can observe them in a very quantitative manner, in a very fast way. And 15% of the disease-causing genes in human is largely conserved in yeast. That is, it can be found easily in fission yeast, and thus the study of those genes in the simple model organism makes it more easier for us to get insight into a complex model organism like humans. And it divides every 3 hours, so that is having due to this rapid growth rate, it is more cheaper to maintain. So on the right is a transmission electron micrograph of S pombe or fission yeast cells. So how do a fission yeast cell divide? So as I told you before, the yeast cells divide similar like a human cells. It involves placement of an actin and myosin based contractile ring at the center of a cell or actomyosin ring at the center of the cell. The ring-like structure then starts ingressing, thus narrowing its diameter. And finally, the cell separates. This mechanism of actomyosin-mediated ring constriction is conserved from bacteria all the way to humans. Here on the right is a time-lapse video of a single yeast cells expressing a ring marker, RLC1, going through cell division. As you can see, the yeast cells elongates in a polarized manner, and in the red RLC1, it assembles the ring. The ring then undergoes constriction, and in the bright field, the cell separates. This is how a wonderful yeast undergoes cell division. And this process can be easily observed in quantitative microscopy, and each and every step of it and kinetics of it can be dynamically controlled and monitored very carefully. So it has been noted and it has been reported like 2 decades back, like 20 years back, that during cytokinesis of animal embryos like zebrafish eggs, frog eggs, and medka fish eggs, calcium concentration increases at the cleavage furrow, or the place at which the cell divides, the center of the cell, where the ring forms. So as you can see from the pointed arrowheads, this dividing cell has been injected with a dye that labels the calcium, and it shows localized elevation of calcium signal at the cleavage furrow. In zebrafish, similar observation has been made in frog eggs, and similar observation has been made in medka fish. So this suggested that calcium transients or spikes were observed during the cytokinesis of animal embryos. But till date, no one knows what is the mechanism of this spike. How does it occur? And what is the importance of this spike? Is it just merely a correlation that is coming that it is occurring during cytokinesis, or this spikes has any role to play during cytokinesis? So I'll switch gears from here and we'll explain a little bit about calcium homeostasis. So this on the left is a single cell showing calcium homeostasis going on inside it. So the intracellular calcium concentration is maintained in a very spatial, temporary, tightly regulated manner, and it is controlled by mostly two mechanisms: influx and efflux mechanism, governed by the channels present on the plasma membrane or the cell membrane, and release mechanism present on the calcium storage organelles like endoplasmic reticulum and vacuole. These two organelles are like another cell organelles present inside the cells, which releases calcium in case the cell needs to survive a calcium depleted environment. So as you can see, the calcium enters from the plasma membrane through ion channels present on the surface of the cell membrane, and then regulates various key downstream regulatory pathways. It can be regulating the downstream key regulatory pathway through in a free manner or in a bound state with any cytosolic protein. This calcium, if excess in the cytosol, can be pumped out of the cells using calcium pumps like plasma membrane calcium ATPase pump or any other uniprot or co-transporter mechanism. Calcium homeostasis is known to play a vital role in many physiological and cellular processes in our body, starting from wound healing to relaying the signals at the synapse of two neurotransmitters, to cell division, to morphogenesis, where it regulates cell polarity, muscle contraction, where calcium is needed for skeletal muscle contraction, hormonal regulation, where calcium is needed, like calcitonin and parathormone, which are regulated by calcium. Structural regulation, for example, bone mineralization is governed by calcium, and cell adhesion, how a cell gets attached and move on its extracellular matrix or a basement membrane. All these are regulated by diverse calcium signaling pathways. But our lab focuses on the role of calcium during cell division, as there is a very limited knowledge on the role of calcium during cell division. Misregulation of this calcium homeostasis may cause, or calcium imbalance in the cell may cause uncontrolled cell proliferation and hence cancer. One of the classical features of cancer is uncontrolled cell division. So we are interested in studying the role of calcium during cell division, as the knowledge is very limited. So currently, the gap in the knowledge includes: is the calcium transient or the calcium spikes conserved in other organisms? What are the function of these calcium transients or spikes? And what are the mechanism of these calcium transients or spikes? For today's talk, I will be focusing on the first two category today. Is the calcium transient conserved in other organisms? And what are the function of these calcium spikes? So the objective of my talk is to determine if calcium spikes are evolutionary conserved features in other eukaryotes. And my hypothesis is calcium spike is conserved mechanism to regulate the constriction of the actomycin ring. Remember the ring that I showed before, which needs to constrict in order for the cell to separate. So my hypothesis is that calcium spike is a mechanism, regulatory mechanism to control the constriction of the actomycin ring during cytokinesis. And the specific aim include to examine if the calcium spike is an evolutionary conserved mechanism in fish and yeast, and to examine the function of this calcium spike. To label intracellular calcium, we perform live cell calcium imaging in our lab, and we use a genetically encoded calcium indicator, which we express endogenously in the yeast cells. It's kind of, by expression, I mean we install this reporter inside the yeast and make them express this. So we use GCAM6S, which is an ultrasensitive probe to label intracellular calcium. So this probe will only fluoresce upon binding to calcium. So this probe is made up of mainly three components: circularly permuted EGFP, which is CpEGFP shown in green, which is a fluorophore; CAM, which is calmodulin, which is a calcium binding protein; and M13, which is another myosin light chain protein, which binds to calcium. So calcium binds to the calcium binding protein calmodulin, and then triggers a very high increase in fluorescence. Normally, the circularly permuted GFP cannot trigger fluorescence because its N terminal and C terminal are covalently linked together, resulting in no fluorescence at all. Only when calcium binds to it, it liberates or releases the fluorophore trapped in the beta barrel of this circularly permuted EGFP, and results in strong calcium-specific increase in fluorescence. So if it fluoresces, that means it is increasing the calcium concentration. So to check, we express this GCAM in our fission cells. So to check and functionally validate this expression system, we first use hypoosmotic shock, which is a known trigger for calcium signaling pathways. So we used different doses of hypoosmotic shock, and we found calcium signaling is triggered in a dose-dependent manner. That means as we increase the dosage or intensity of the hypoosmotic shock, we found the intensity of the calcium spikes or transients to increase sharply. Here on the right is a time-lapse video of multiple population of yeast cells undergoing hypoosmotic shock at 1.2 molar sorbitol stress. So once the time zero appears, from -30 to time zero marks the start of or the beginning of the hypoosmotic shock. So let's see what happens at time zero. As you can see, the cell undergoes hypoosmotic shock and results in strong increase in calcium transients or spikes. I'll play the video once more. And as you can see, as cell reaches hypoosmotic shock, there is an increase in calcium spikes within population of cells. And this generalized nature of this calcium spike has been observed in all the population of cell, and there is no single cell heterogeneity. And this observation was found to be dose dependent, and we can monitor it using single cell quantitative microscopy. Next, to identify the calcium spikes, next to employ this reporter and identify the calcium spikes during cytokinesis, we imaged the yeast cells during the entire phase of the cell division. Normally, it takes 3 hours for the cells to divide. So we expressed cells co-expressing GCAM and a ring marker in red, GCAM in green, and looked for calcium spikes during cytokinesis. We observed that as the cell approaches the period of ring constriction, it is marked or accompanied by calcium spikes. So here is a time-lapse video on the left. And as you will see, when the time zero approaches, which marks the start of the ring constriction, it is accompanied by a strong calcium spike. So as time zero approaches, the cell shines brightly, which is marked by increase in calcium spike. So I quantified this for multiple cells in the 3D plot. The y-axis is the GCAM fluorescence, which is intracellular level of calcium, and the x-axis is the time for the ring constriction. Here, -30 to 0 represents the ring assembly period, and 0 to +30 represents the ring constriction period. We found that calcium spike was not there during the ring assembly period, but was far significantly increased when the ring started to constrict. So we termed this spike observed during the constriction as constriction spike because it was found during the ring constriction period. Next, we found another spike that was attached or following the cell separation. Here we imaged the cells expressing GCAM during cell separation, and we found calcium spike was similarly accompanied during cell separation. here is a time-lapse video, and as time 0 is marked by cell separation, you can see physical separation of the cell breaking up. At time 0, the cell breaks up and is marked by calcium spikes. Again, on the y-axis of the graph, 3D line plot is the GCAM fluorescence, which is calcium, and on the x-axis is the time 0 before and after the cell separation. Time 0 is the start of cell separation. And again, we found increasing intracellular calcium spikes during cell separation. So till now, we have found 2 calcium spikes accompanied during the cytokinesis of fission yeast. We name this spike as separation spike. So the 2 calcium spikes that was found is constriction spike and another one separation spike. Next, we wanted to check whether these spikes that occur during cytokinesis is due to influx of calcium from the outside or extracellular environment, or release mechanism triggered from inside the cells. Whether calcium is influxed or whether calcium is released from inside the cell. So we treated the cells expressing GCAM and a ring marker with EGTA. EGTA is a calcium chelator which scavenges calcium molecule inside the cell. It's a divalent calcium chelator. So we treated the cells expressing GCAM and monitored the calcium in the presence and absence of EGTA in a dose-dependent manner. So on the x-axis is dose-dependent, untreated 0, and 1 millimolar of EGTA and 2 millimolar of EGTA. And on the y-axis is the intracellular calcium. We found that treatment of EGTA strongly results in decreased or depleted calcium spikes, suggesting that the calcium spikes occurring during cytokinesis was partly due to influx of calcium from the outside environment. So here is a time-lapse video where time 0, there is no calcium spikes in the cells. I'll play the video again. In time 0, there is no calcium spikes in the cells. So to ascertain that more and to verify the result, we transiently depleted the cells with external calcium. So we depleted the cells of external calcium and grown the cells in a calcium-free media. And we observed similarly that there was no calcium spikes during cytokinesis, suggesting that the calcium spikes occurring during cytokinesis is a result of influx of calcium from the outside environment and not due to release. So we wanted to check what is the fundamental importance of this calcium spike during the cytokinesis. Does it play any role? Does it have any molecular importance? So we treated the cells with EGTA and monitored the ring constriction progression. So we monitored the rate at which the contractile ring starts ingressing and found out that the speed or the velocity at which the contractile ring starts ingressing slowed down upon treatment of EGTA. So remember, EGTA is a calcium chelator. So chelation of calcium slowed down the ingression of the contractile ring, suggesting calcium spikes control the cleavage furrow ingression. As you can see on the left graph, the rate is on the y-axis, the rate at which the contractile ring starts constricting. And on the x-axis is the dose dependency of EGTA. On the right is a time-lapse micrograph of single cell going through cell division in the presence or in the absence of EGTA. And it shows that in the presence of 2 millimolar EGTA, the cell took far longer time to constrict. The assembly time of the ring assembly time was normal, but the constriction time significantly altered. So it took longer for the cell to constrict. Because it took longer for the cells to constrict, the speed at which the ring constrict has decreased. So we did the same thing in a calcium-free media and observed a similar result, that ring assembly was same, but the ring constriction is severely delayed because the contractile ring was ingressing very slowly. That's why it is delayed. This suggested that calcium spike controlled the ingression of the cleavage furrow. Next, we wanted to check what is the importance of the calcium spikes during cell separation. Remember, we found a calcium spike accompanying the cell separation. But what is the importance of this calcium spike? So we treated the cell with EGTA and we found 35% of the cell lysed. suggesting that depletion of the external calcium led to frequent lysis of the daughter cell post-cell separation. That means they die. 35% of the cell die during treatment with EGTA when calcium molecule is scavenged. So we go back to the question we raised earlier. Is calcium transient conserved in other organisms? Yes, calcium spike are conserved in Schizosaccharomyces pombe fission yeast, which is a unicellular eukaryote, unicellular single cell eukaryote. What are its intracellular function and roles? At least till now we found two roles. The calcium spike controls the constriction of the actomycin ring because it decreased the rate at which the ring constricts when we depleted the spike. And it maintains the integrity of the separating daughter cells because when we depleted the spike, 35% of the cells lysed. Thus, conclusion is calcium homeostasis plays a conserved role in regulating cytokinesis. In my acknowledgement, I would like to thank my PI, Dr. Kian Chen, and my lab members and graduate and undergraduate member, and our funding resources, National Institute of Health, General Medical Science, University of Toledo Startup, and D. Archcock Memorial Fund. Thank you. With that, I would like to take any question, if you have any.
Thank you very much, Abhishek. That was a wonderful talk. That was really interesting, and I actually understand now how, at least to an extent. I'm still a chemical engineer, so I gotta, my biology background's a little bit weak, but I followed that really well. So that speaks highly of your communication skills.
Thank you.
All right, so we will just go ahead and jump straight into the questions. For anybody in chat, if you have questions, if you're thinking of them right now, certainly ask them. I've got I'll get them queued up. So first, we got a question here. Okay, so you talked about that the fission yeast had about 15% of the genes or disease-causing genes can be found in fission yeast.
Yes.
What types of diseases are associated with those genes?
There are many. Like there is one gene called PKD2, which is polycystic kidney disease gene, PKD2 gene, which causes polycystic kidney disease. Oh. And there are several other genes, as far as I remember, which causes several defects during cell division. And I can't remember at the back of the mind, but
sure.
Yeah, there are several genes that causes disease. One of that could be polycystic kidney disease, which is kind of an end-stage renal failure. And it's very common in America. Almost 1 in every 500 to 1,000 Americans are affected by that.
Interesting. I did not know that.
Yeah. That gene is called PKD2 in humans, and we have that gene also in yeast.
Interesting. Okay. So, I guess this might be a dumb question. Do we know why?
So PKD2 is an ion channel, which is thought to be influxing calcium. So this disease is thought to cause due to misregulation in calcium signaling.
Okay, all right, that's interesting. I never knew that. That's actually really cool.
Yeah.
Okay, so you talked a little bit about, obviously you talked about the calcium chelator, that you use to sort of establish, whether calcium actually had a role in this or not. So this question came before you asked that, or before you showed that, but I think it's still probably worth exploring a little bit. Excuse me. Is there a minimum level of calcium that's required for the division that you showed to occur? And if there is, I guess, well, you kind of answered this one already, but I'll go ahead and ask the question. You can speak on any part of it that you would like to. If there is a sort of a minimum level, what happens below that level?
Yeah, that's a very good question. We don't know what is the threshold or the minimum level of calcium that is required for cell division. Mm-hmm. For when we depleted the external calcium completely, although we depleted for a transient period of time because we hypothesized that cell cannot survive without the calcium. But when we depleted the entire calcium, that's a 0 millimolar calcium. And we found the defect is not that much great. So it decreased the furrow ingression, but the cleavage furrow or the contractile ring still ingressed, but the rate at which it ingressed slowed down, but it still ingressed. That means calcium contributes to the contractile ring ingression, but it does not play essential role in it. So we do not know what is the minimal level of calcium that is required. And of course, the misregulation part, if there is misregulation of this channel,
Mm-hmm.
the yeast cell does not divide. I can only talk about yeast cells, but
Yeah.
if I translate that to complex human, there are several diseases that occur due to misregulation of calcium signaling, like Alzheimer's, Parkinson's, polycystic kidney disease.
Oh, I
Yeah.
didn't realize that Alzheimer's and Parkinson's were related to calcium
Yeah, I mean,
regulation.
calcium contributes to it. Alzheimer's and Parkinson can be regulated by many other factors
Mm-hmm.
or many other signaling pathways, but calcium could be one of the signaling pathways.
Oh, okay. So the work that you're doing here kind of has a lot of a wide ranging application, then, at least potentially.
Yeah, yeah, definitely.
Okay, that's yeah. I'm learning a ton right now. So you talked about hypoosmotic shock. So
Yes.
that's not something I'm really overly familiar with, and probably at
Mhm.
least a few people in chat aren't either. Could you talk a little bit more about what that is?
Yes. By hypoosmotic shock, I'll go back to the. So hypoosmotic shock means I'm decreasing the osmolarity of the medium.
Mm-hmm.
By osmolarity, I'm meaning I'm decreasing the solute concentration of the media.
Okay.
So that the cell senses that as a stress. So
Mm-hmm.
in hyperosmotic shock, that is when you add something or add solute to it, is called hyperosmotic shock.
Mm-hmm.
So when you add some solute to the media, the cell shrinks
Okay.
and the cell lysed.
Okay.
In hypoosmotic shock, the cell swells because of more solute in the cells. The
Mm-hmm.
cell swells, and that triggers rupture. The cell swells and bursts. So cells
Mm.
are intelligent species because in response to hypoosmotic shock, because cell swells, cell don't want to burst. So
Yeah.
what they do is they activate the ion channel on the surface, results in calcium influx, redistributes the ionic homeostasis that recovers their volume, and then they survive.
Mm.
So
Okay.
calcium signaling is basically a response to the hypoosmotic shock. As you can see, in the bright field image, I will play the movie again. Can you
Sure.
see the movie?
Mm-hmm.
The cell will swell at time zero.
Oh, okay.
Here, at time zero, the
Yeah.
cell will swell at time zero. Here,
Oh, yeah, sure did.
yes. So cell will burst. So cells are intelligent species. They don't want to burst. So they
Yeah.
will activate ion channels at the surface and let in all the ions come in to redistribute them and balance the ionic homeostasis so that they can recover the volume. They don't want to burst.
Okay. Interesting. So what's the like in a person what's the equivalent condition in a person like in real life? Obviously, you could do something similar with human cells,
Yeah,
but
we don't. Sorry, as a human, we have a tissue level organization.
Mhm.
So you can think when you bath, when you take bath in a lot of water, you have
Mhm.
numbness. So that's hyperosmotic shock.
Mhm.
So kind of like it trinkle. There's numbness in the finger,
Right.
or
Okay.
when there is rain and the bacteria is there in the soil. The rain to the bacteria on the soil. It's a hypoosmotic shock. So the
Ah.
bacteria activates many signaling pathways to survive. Otherwise, they will burst because it's a well-known phenomena that in response to hypoosmotic shock, the cell swells, and in response
Yeah.
to hyperosmotic shock, the cell shrinks.
Interesting. That's really fascinating. It's impressive how quickly it does that.
Yeah.
Okay. So, what? I'll go ahead. Anybody in the chat that has? Oh, there we go. We got an extra question coming in. Let's see. So, using the calcium chelator, you're depleting the external source of calcium. But what happens with the internal reservoir? Is it not enough to compensate during cytokinesis, or is it not released at all without the external input?
That's a very good question. So that's why to verify the result, we have to deplete the external calcium completely. So EGTA, we do not know whether it is cell permeable or not, and whether it titrates out only the external calcium or not. So there is no literature whether it can go inside the cells and percolate through the cell membrane and titrate out the internal calcium. So we are doing experiment where we control the release mechanism and block the release, and then see what happens to the calcium spike. For now, I can say that the calcium spike is governed partly by, definitely majority of the calcium spike is definitely influx, but it can be released to a small percentage. Yes.
Oh, okay. You said that you're working on characterizing the internal release right now.
Yes.
Oh.
What happens to the calcium spike if we block the internal release? There are
Ah.
certain pharmacological inhibitors that we are using to block the calcium spike and in the internal release of calcium, and then check if the calcium spike is there or not.
Interesting. Okay, I'll be curious to. Yeah, that's a great way to check it. That's really cool.
Yeah.
Um, I had a question and I completely forgot what it was. It slipped my brain. So I'll try and think of it. If anybody else in the chat has questions that they want to ask, please feel free to get them in real quick. Oh, I know what it was. So well, you kind of partly answered it with what you just said, but what are the future directions of this work? So you're working on the internal calcium regulation
Yeah,
right
the
now,
future
but
direction of the work is a long thing. I did not spoke about the third part. What is the mechanism?
okay, okay,
How it is regulating.
okay,
So we think there may be ion channels, but how many ion channels? There are so many ion channels.
mm-hmm.
So we have to pickpocket like which ion channels we have to select. There are so many ion channels on the surface of yeast. Like
Mm-hmm.
we have to select which ion channels to look for, and then we can progress from there.
Okay.
If
So
mediating.
how do you do that? Like, what's just generally speaking? Like,
Yeah, so we can do a screening,
okay.
like a broad screening of
Mm-hmm.
all the ion channels in yeast. The screening is particularly very easy, and check like all the ion channel mutants and see which is responsive to calcium signaling. Like we can grow them on a plate and see which
Okay.
mutant die or which mutant survives.
Ah, okay. All right. So it's pretty straightforward. Just takes some time.
Yes.
Okay. Can you talk a little bit about how you're going to try and get at the mechanisms? Again, just generally speaking, what like
Yeah.
you
So
select the
yeah.
ion
So
channels and
we
then
would like to look into the ion channels. What is the mechanism? Means how the spikes are regulated. So we
Mhm.
hypothesize that there may be ion channel on the surface,
Okay.
or it might be there may be pumps that is releasing the calcium from the
Mm.
internal store.
Okay.
So we exactly have to determine uh, which which channel
Mm-hmm.
could be regulating. That is a part of the mechanism.
Okay.
Which whether it's an influx or efflux or release mechanism.
Gotcha.
That is the mechanism, like how it is controlled. All these
Mm-hmm.
calcium spikes.
Mm-hmm. Okay. Are there potential what's the word? Not conflating. Are there other effects that like so I see for instance that you have like ATP is used to pump the calcium in and out. Are
Yeah.
there
So those are
other
yes. So those are ATPs. So those are pumps. Pumps means ion channel means these are passive gradients. So it doesn't
Mhm.
require any energy. So the calcium can
Oh.
easily flow. But pumps means they need energy from the production of ATP, from the utilization of ATP,
Hmm.
and they convert that to ADP to pump calcium out. So they spend energy. So pumps spend energy to pump out any extra intracellular solutes outside the cells,
Hmm.
while ion channel does not spend any energy to pump in
Okay. Okay.
solutes.
Can the ion channels be what's the word? Can they be sort of and they can be blocked because you're that's some of the stuff that you're looking at. But
They can be
can
blocked
they be
easily.
blocked in a natural way? I don't know how to put this the right way. But so again like in a normal person without the
Mhm.
pharmacological uh
Hmm.
whatever methods that you're using to purposefully block them. Are
They
there
can
are there
be they
naturally
can be either
occurring?
blocked. They can be either blocked during mutation. For
Hmm.
example, there is a disease called CFTR, cystic fibrosis,
Hmm.
that is caused due to a blockage of or mutation of the ion channel called CFTR,
Mm.
cystic fibrosis transmembrane ion channel, as far as I remember.
Okay.
So there is mutation in that ion channel that causes the disease. So there is two ways to block: either use pharmacological inhibitors to
Hmm.
block, but the pharmacological inhibitors can block many other ion channels. So it's like
Right.
a chemotherapy. It not just kills normals, it not just kills cancer cells, but also kills many normal cells. So pharmacological inhibitor is non-specific. That means it can block a group of ion channels, like
Mm.
several family of evolutionary conserved and sharing similar feature of ion channels, like trip channels or mechanosensitive ion channels, like that. But
Okay.
best way would be to genetically mutate them and block them.
Got you. Okay. That's really cool.
Yeah.
It's interesting to learn about the techniques that are available to you to do that kind of work.
Yeah.
That's really neat.
Yeah.
So if there's any final questions, go ahead and get them in. I'll go ahead and start wrapping up. But like I said, if a question pops up, we'll sidebar and tackle that real quick. Abhishek, thank you very much for coming on and presenting your work. Like I said, it's really interesting, and you presented it in such a way that even somebody like me who is not at all familiar with the field was able to
No, thank you
follow
for inviting me.
along.
It's,
And
I can improve my communication, so
oh no,
that's
you seriously,
a
you did a
great
great.
opportunity for me too. Thank you for inviting me.
Yeah, absolutely. You seriously, you did a great job. Like that, like I said, I understand. I understand what you're doing, even with all the with all the other stuff that I don't understand. I get what you're doing, why you're doing it, and what you're looking for when you do it. So
Yeah.
that, like, great job on that.
Yeah.
That
Thank
was
you.
very, very well done. It looks like that's all the questions we've got. So we'll go ahead and wrap up. Abhishek, if I could get you to hang on for just 1 second, we'll talk very briefly. And everybody else, thank you all for coming out. We will be back next week with. We have Dr. Carly Ann York from Lenore Ryan. She's a biologist, and we're talking about some of her work. And if you missed it earlier, I'm going to upload the announcement video for the Science Palooza 24-hour stream. Like I said, that'll be July 2nd, and I will also be posting, of course, the schedule and or the available schedule times and the sign up survey for it, and we will try and get that going. But for now, everyone have a great weekend. Please stay safe. If you haven't gotten vaccinated yet, please do that. And I will hopefully see you all next week. I hope you have a great weekend. Bye, everybody.